Hi, my name is James Brown. I'm with Aldevron, and just to make sure you're in the right place, this is the Gene-Based Medicine Development Workshop, and this is session 1, Gene Editing, Overcoming the Technical Challenges of CRISPR. And thanks to everyone for arriving at 7:15 a.m. This is the third time that I've been involved with this, and I'm always impressed with the people who make it this early in the morning. I'd like to thank ARM for organizing the meeting on the Mesa. It's always been a great meeting with a lot of collaborations going on, a lot of good science presented. So we've done this workshop in a couple different formats, and what we think works best is really no slides and just have a discussion with some introductory questions, and then it is a workshop, so we expect you to work and ask questions, and we've got this clever little thing that you can toss around that's the microphone that's a lot of fun. So I'd like to start just by introducing myself and the panelists here and have them talk a little bit about their companies and the progress they're making, and then launch into some questions, and then have the audience participate here and have a good discussion on the technical challenges of CRISPR. So as I said, my name is James Brown. I've been with Aldevron for about 3 years. Prior to that, I was at Regenexx Bio, which is an AAV gene therapy company. I've been in biotech for about 20 years. Aldevron is a contract manufacturing organization. We're pleased to serve the industry with a variety of products, including Cas9, even at the GMP level grade as an off-the-shelf product. So I'd like to start off with James Burns. Jim joined Casebia in November 2016, and they're focused on developing new therapies based on gene editing for hematological, immune-mediated, and ophthalmological and cardiovascular diseases. Prior to that, he was head of Sanofi's Boston Research and Development Hub, and then the Sanofi Genzyme Research and Development, and head of Sanofi North America Research and Development Hub. And Jim joined Enzyme and built a biopolymer research and development group that led to the commercialization of several products that became the basis for biosurgery business. So welcome, Jim, and maybe you can tell us a little bit more about your time at Casebia and the progress you're making. I know that in the discussion we were just having before, it's all about space and expansion, which is good news. So let us know how things are going.
Yeah, thanks, James. It's a double-edged sword. You need more space, but we're in Cambridge, so it's also really expensive, so it does a lot for your burn. But first of all, very happy to be here and happy to talk about Casebia and happy to talk about gene editing as well. So we're a research stage company. We were launched a little bit over 2 years ago. I was employee number 3, not quite 2 years ago. And we have a very unique model, so I just want to just very, very briefly speak about the model, because it's a joint venture between Bayer and Crispr Therapeutics, and Bill was very prominent early on in starting Casebia. And the idea was to try to infuse a significant investment, $300 million. It's a very large Series A, as well as foundational intellectual property, and to set up an independent organization. So even though it's a joint venture, it actually is run independently. So I report to the board. So Bill was effectively my boss, but we ran things independently, and we also were able to leverage learnings from CRISPR as well as from Bayer to try to help jumpstart our programs. We're focused mainly in blindness at this point, as well as non-malignant hematology and autoimmune diseases. So we're a research stage company right now, but we're rapidly advancing towards development candidates, which we hope to have more than one of those next year.
Thanks. We're also joined by Stacey Cohan, VP of Business Development at Editas. She joined Editas in 2017 with more than 20 years of experience in corporate and business development and life sciences. And prior to that, she worked at Genzyme and Sanofi at corporate development and business development roles, and most recently served as global head of rare diseases and business development at Sanofi. She's led all facets of strategy, business development, licensing, et cetera. And she currently serves as a member of the board of trustees of the Huntington's Disease Society of America. So welcome, Stacy, and how are things going at Editas?
Thank you. Thank you for having me. They're going great. So I joined Editas about a year ago, And I'm just so excited to be in this rapidly evolving field. Certainly lots of activity in terms of deal flow. There's lots of activity in terms of scientific advancement, company formation. So it's a really great place to be for a business development executive. As a company, we're 5 years old now, and we're at a very important moment, both in terms of taking our company to the next stage of growth and also shaping the field. This month, we'll file our first IND, and that IND represents the first in vivo CRISPR medicine to enter patients anywhere in the world. So we're pretty excited about it. We're looking forward to taking that next step and advancing the program. And then behind that, we've sort of built an array of gene editing based ocular development candidates. And we also are investing heavily in cell therapy now, where we think that gene editing has the potential to create transformative medicines in many different indications, including the ones that we're working on, sickle cell disease, beta thalassemia, and cancers.
Thanks. We're joined by Rachel Haurwitz, President and CEO of Caribou, and she was the co-founder of Caribou and has been in her current role since its inception in 2011. Her research background is in CRISPR-Cas biology and is also co-founder of Intellia Therapeutics. In 2014, she was named Forbes magazine 30 Under 30 in Science, and 2016 Fortune 40 Under 40. I don't know if there's a 50 Under 50, but let's go for the trifecta. In 2018, the Association of Women Science recognized her with the annual Next Generation Award, and she's an inventor on several patents and patent applications covering multiple CRISPR-based technologies. So welcome, Rachel, and tell us how things are progressing at Caribou.
Good morning. Thanks for having me. For those of you who don't know the Caribou story, I'll give you just a little bit of our history. So as mentioned, we're a little more than 6 years old, co-founded by Jennifer Doudna, Martin Yinick, and other CRISPR pioneers. And we really started as a platform technology company with a broad focus on deploying CRISPR in any market with bio-based products. And so through partnership, we've worked everywhere from human health and drug discovery to animal breeding, crop breeding, and industrial applications. And along the way, we've actually developed a new flavor of CRISPR technology that has really improved specificity. And with that in hand, we're really focused now on advancing our own internal pipeline of human therapeutic products split across two key areas. One is immuno-oncology and specifically gene-edited cell therapies. And the second is modifying the microbiome with gene editing technologies for specific therapeutic outcomes. We're also a research stage organization and really excited about the progress that we're making and that the whole field is making and how incredibly rapidly this space is moving.
Thank you very much. We're joined by Jennifer King, Senior Vice President Business Development at Intellia. At her current role, she's responsible for accessing external sources of innovation and establishing key partnerships. She brings 20 years of experience in the biopharmaceutical industry. Prior to her role at Intellia, which she joined in June 5th, 2015, she held position Senior Director of Business Development at Shire, where she was responsible for sourcing and evaluation, negotiation of licensing and acquisition opportunities within the company's corporate development group, and also worked in Shire's Rare Disease and Commercial Organization and Director of New Products. So welcome, Jennifer, and how are things at Intellia?
Well, thanks. I'm delighted to be here. Things at Intellia are moving very quickly and very rapidly and are very good. As Rachel mentioned, we actually are formed from IP that we licensed from Caribou back in 2014. And during that time, we have rapidly grown from a small company when I joined, sub 50 in 2015, to now over 200 folks focused primarily on therapeutic applications of CRISPR-Cas9 in the ex vivo setting as well as in the in vivo setting. We are working very closely with our current partners. So we are partnered with Novartis for ex vivo applications of genome editing in hematopoietic stem cell biology, as well as supporting their CAR T therapeutics, their next generation CAR T. And Intellia also has its own programs in immuno-oncology, in gene-edited T cells for immuno-oncology, with our lead indication being AML. In addition, we have a very active program in vivo applications of genome editing using a proprietary non-viral delivery mechanism, chemically synthesized lipid nanoparticles. And we are partnered with Regeneron on applying that LNP and CRISPR technology in the liver. And we're also expanding beyond the liver to other organs where we think the LNPs have promise. And as being formed only in 2014, only 4 short years ago, we are rapidly moving towards the clinic, and we expect to file our first IND towards the end of next year.
Thank you. Last but certainly not least, we have Bill Lundberg, who was the Chief Scientific Officer and first employee of Crispr Therapeutics, establishing the company in Cambridge and growing CRISPR through its first clinical trial application, public offerings, and financing. Prior to that, he was head of translational medicine at Alexion, where he led research and development activity from discovery through early clinical and regulatory. He was also the chief medical officer and director of Intellia Therapeutics, which was acquired by Alexion, and currently director of several privately held biotech companies and 2 nonprofits. Bill, you got a broad view of the industry there. What do you see?
Yeah, thanks, James. First, to correct one thing, I was the first U.S. employee when CRISPR was brought to the U.S. I also wanted to thank you and thank my panelists and thank ARM for inviting me to participate in the panel. I think it's really tremendous. Let me say just a quick few words about CRISPR Therapeutics, and then I'll comment on the field. CRISPR Therapeutics was developed contemporaneously with the other companies on the panel, essentially. And we also are focusing on applying CRISPR-based medicines or CRISPR-based approaches to developing medicines for human therapeutics. Our lead programs, for which we filed clinical trial application and INDs, are in hemoglobinopathies and particularly sickle cell disease and beta thalassemia. We have a development program using CRISPR technology to modify CAR-Ts for therapeutic benefit, as well as a series of in vivo programs. We have 2 key partnerships with Vertex in our hemoglobinopathy programs and with Bayer in setting forth Casebia, which have been really instrumental in us furthering our own abilities and capabilities. And that's really been tremendous and great fun. I also want to say, just in terms of the field, it's remarkable to sit up here with my colleagues to think about the billions of investment that have been made in not only endonucleases, but specifically RNA-guided endonucleases across many, many different potential therapeutic programs. It's created multiple companies, over 600, 700 jobs in these companies here alone, and with the opportunity to make some really important medicines for patients. I mean, the past 4 years has just really been tremendous. And so, hats off to all my colleagues here and to all of you working in this field to help move this forward. So hopefully we can make some really important medicines.
Thanks to everyone, and I'm impressed and humbled by the group up here. It's really exciting to see all the progress being made. But the title of the workshop is Overcoming Technical Challenges, so I think everyone recognizes that there's a lot of progress that's been made, but still things out there that need to be overcome. So we just start off with a question here. The biology of CRISPR can be complex with many challenges, including efficiency and off-target effects. So for the panel, I would ask, what challenges are you facing with your programs? How do you address them, and how do you evaluate the advances in CRISPR being made and decide which are worth pursuing? There's a lot of academics out there and small companies. So Stacy, maybe you can help us answer that question and start off.
Sure. Yeah, so I think, essentially development of gene editing based therapeutics is not fundamentally different from what others have done in cell and gene therapy, and maybe in a broader sense with biologics more generally. Having said that, though, the one thing that really stands out in terms of detailed analysis of on-target and off-target editing, that could be the one area that really has required all of us to create new technologies and think hard about how we're going to assess that for the regulators and essentially for the patients. We're glad to see that the regulators are endorsing an approach that we've, I think, collectively even advocated for essentially requiring multiple orthogonal approaches to looking at on and off target edits. And I can just comment on like an example of one of the technologies that we've developed. It's called Editas. It stands for Unidirectional Targeted Sequencing, and essentially it's a novel sequencing methodology that allows for detection and quantification of on and off target editing, insertions and deletions, also translocations and other structural changes. So it's an approach that we will incorporate into each of our programs, and I think there's a host of other things that all of us do to try to ensure that we have a very good understanding of where our products are modifying the genome.
I have a quick question. Is that something you developed internally, or did you license from a group or academic, or?
We license that internally.
Okay.
But I think that's a nice segue into the second part of your question, which is about how we think about CRISPR advancements. So I think being a leader in gene editing, certainly we have tremendous visibility into the types of advancements that are being made. And so we take each one of those developments case by case in a way. We've worked to build our platform over time and certainly make sure that it's robust enough that we can develop drugs. So we've done that through internal innovation. We've also used business development and transactions to access things. So I can give a couple examples there of things that we've found important enough to make that investment. But essentially, it's on a case by case, and we're looking at the relative merits of any given advancement and the application to our portfolio and that sort of thing. But a couple of things that we thought were worthy of investment are one is around the panel of genetic or of CRISPR-based enzymes that we have available to us. CPF1 is a good example of that, or it's otherwise known as Cas12a. So in addition to CRISPR-Cas9, we have the CRISPR CPF1 system operating in-house. And that system essentially, it's a structurally distinct protein, but it gives us acts similar to CRISPR-Cas9, but allows us to make edits in more sites within the genome. So it gives us sort of a broader spectrum of opportunity, I guess, to edit the genome. And so we utilize that as a method for getting the most specific edits that we can get that achieve our intent. And we took license to that technology a couple years back. We have the exclusive license to use it internally at Editas. And then there's another piece that comes to mind, which is around what we think is very important, the RNA, guide RNA engineering and manufacturing piece. And so we announced an acquisition of a company earlier this year called I2 Pharmaceuticals. I2 was a partner of ours that we were working closely with and had proprietary expertise in RNA synthesis. And so with that company now integrated into Editas, we have readily available high-quality guide RNAs for each of our programs. So those are a couple of examples of areas where we thought it was wise to invest and continue to do that, both build things internally and access things externally. I'm sure my colleagues up here do the same.
Yeah. That's great. Anyone else? Comment?
I think it's a really exciting time, right? The patents on CRISPR were filed earlier, 2012, 2013. And so this technology is very young and very hot. And so you have a lot of academic groups really working on different aspects of CRISPR itself as well as the technology. So it's almost like we're crowdsourcing solutions for potential products. So I think to your point, both development of technologies in-house, but also allowing the environment, the academic environment and small companies to develop solutions as well is just really amazing in this field.
Certainly.
Maybe I'll pick up on something that Stacy mentioned, because I think it's probably obvious to everybody sitting up here, but may not be obvious to everyone in the audience. there's a lot of talk about off-target effects and what that means. And I think most people think about, for a given Cas9 guide complex, you want an edit in one place, you don't want changes elsewhere. And yes, of course, that's a piece of the puzzle, but there are a lot of other genomic implications of genome editing, and especially as we start to look at ever more sophisticated gene-edited cell therapies where there are multiple changes to a given cell, you run the risk of chromosomal translocations or other large-scale rearrangements. So I think it's really important to highlight that I think all of our companies are working really hard on a variety of tools and ways to try to assess that, because it is a lot more complicated than even just the simple sort of classical off-target effect. But I also think it's important that we have a shared vocabulary for that. And so a lot of the organizations in this space, both companies and academic groups, are coming together under the auspices of NIST and in other ways to try to build a common lexicon, a common vocabulary, so that we can all, for our own individual therapies, have a shared way of assessing them and a shared way of discussing that with regulatory bodies, which I think will be really important moving forwards.
That's great. I don't know, do we have that cube thing that we can toss out? Okay. If there's any questions from the audience on the topic, just raise your hand and we'll toss you the microphone. Yeah, we have one here, gentlemen. Yeah, so I think you have to like talk into it or something.
Can you hear me? Am I on? There we go.
Yep, we got you.
You talked about the guide RNAs and you talked about the proteins, the Cas proteins. Can you talk a little bit more about the delivery schemes that the groups are using? Is it viral, non-viral? You mentioned some non-viral up there. Mm-hmm. Can you talk more about the viral schemes that you might be using? And what challenges you might have there?
I mean, maybe I'll comment on that. I've always said that this field is going to succeed or fail because of delivery. I'm sure there's other reasons why as well, but to me, that's one of the real key components. We have to be able to get what we're trying to deliver right where we need to and do it faithfully. And if you're working like we are, and I think all of us are on ex vivo as well as in vivo approaches, then you need a whole toolbox of tools to be able to deliver. So just like my colleagues here, we're looking at different ways of doing that. So for in vivo, we're looking at viral as well as non-viral delivery systems, and we're also trying to look at transient delivery of the Cas9. And we think that's really important for a lot of reasons, just to try to make as safe a product as possible. So that's something we're investing quite a bit in. For ex vivo electroporation, I'm sure all my colleagues up here use that, and I think it's very effective. You have transient delivery when you use electroporation for the Cas9, and you get very high editing efficiency. So what I hope happens is that we continue to invest not only in understanding off targets and how to really characterize that aspect of gene editing, but also invest in new ways of trying to deliver site-specifically with viral as well as non-viral delivery systems. It's something that we're spending a lot of time on, a lot of investment.
And I would add, it's an important point that the need drives the delivery method, not the other way around. We don't approach this and say, oh, we've got a clever AAV pseudotype, we're going to look for an indication. It's really the other way around. For example, I think if we all had a solution for delivering CRISPR tools to 90% of muscle, we'd be really interested in muscle-based monogenic diseases and the ability to fix them, because that's currently the limitation in many of those diseases. But it's really the indication that drives the selection of delivery method.
Yeah, I appreciate that question. I don't know if reading our mind, that was the next topic that we were going to cover. Is there any other questions? And maybe some of the other panelists can comment on these different ways of delivering components to cells, including, as you mentioned, transfection and vectors, and what are some of the options you've considered and advantages and disadvantages?
So Intellia uses lipid nanoparticles for our in vivo work. Again, this is a transient delivery system that we think is really effective. And there are approved therapies using similar LNPs to deliver nucleic acids to the liver. But I think as Bill mentioned, they deliver great to the liver, but how do you get it beyond the liver? And, is the delivery system limiting the indications you can go after, or, how do you balance that?
Any other questions? We got a couple. Gentlemen in the back, one. Can you toss that over to? There you go. Thank you.
Shout anyway.
Yeah, there you go.
So my background is in integrating retroviral vectors, so I've sweated plenty about the liabilities associated with that. So my question to you is, when do you stop worrying about off-target effects? When is enough enough? Because there are already plenty of indications that show that quite a lot of chromosomal damage can be managed within a healthy system. So how do you know when to stop? Or how do you know when not to go?
Maybe I'll take a crack at a piece of that. So I think part of it is the context of what you're doing, and obviously specifically the unmet medical need and what options patients do or do not have in that therapeutic context. And so treating a rare form of cancer for which patients have little to zero options and a very short timeline without treatment, that's a very different context than treating, say, a genetic disease where the patient is not in a situation of being at risk of losing his or her life in a short period of time. And so I think a lot of what we're seeing in the field is people are appropriately chasing after indications with severe medical unmet need, asking a lot of these questions intentionally and unintentionally along the way to get a better understanding of this. I do think it's also important to point out that simply understanding off-target effects at the genetic level is only a piece of the puzzle. It's a really important piece of the puzzle, but it's only a piece. And understanding the functional consequences are equally important to understanding and assessing risk and safety for any given therapy. We've seen this in the gene editing field before, right? This is not new. Even the very first therapy that Sangamo put into patients years ago at this point had a very well characterized off-target gene editing effect. And they looked at this very carefully, very rationally, and in discussions with the FDA got comfortable with that specific off-target effect and understanding what its risk profile might be. And so I think we already see that the field is trying to be very thoughtful about this, but I also think it'll change over time as the technologies to assess this only get better and better. What we can measure today in terms of off-target effects is orders of magnitude better than what we could measure 2 years ago, and no doubt 2 years from now will be a different world as well.
It sounds like it's a risk-based approach. You mentioned Sangamo. Do you in general find that the regulatory agencies are comfortable with that and willing to tolerate a little more risk for some of these more dire diseases?
I would say that the agency's approach generally is pretty standard, whether it's the FDA or the European authorities or on the national level in Europe, or the European-wide level in Europe. And that the issue really is, as Rachel has said, a risk-benefit consideration. What is the potential risk of the therapy? What's the potential benefit? And what's the context in which we're putting this? I mean, I don't think we ever stop worrying, right? We're putting something into a person. So we're always going to worry about that. But we want to have as much confidence as we can that the risk-benefit ratio to the patient is likely to be favorable.
Great, thanks.
Yeah, I would just add that in
Sure.
our regulatory dialogues as well, we've seen agency being, sort of very cooperative and thoughtful about how we do this. And I would echo your comments that it's very, it's just very important to have that risk-benefit profile mapped and understand what we think is going to happen. But they're interested in helping us advance this technology. And there's not some distinct fear around genetic editing per se. It's more about just product-specific considerations and the profile of that product.
You know, we haven't gotten to that point yet that you all have gotten to in terms of the regulators, but it does appear to me that they're willing to work with us to really try to understand the balance between risk and the benefit for the patient, especially since this is potentially such a powerful tool where you can actually cure diseases. And one question I had for you, Bill, is what has been your experience on how the European authorities view this relative to the FDA? Do you see that they're on the same page? What are the differences? What are the similarities there?
Well, I mean, this applies across therapeutic areas, not only to ATMPs or complex medicine, cell and gene-based medicines. There's inherently differences in view. Some regulators want you to do one particular type of preclinical study. Other regulators reject that outright and expect something entirely different. And sometimes you can bring them together, and sometimes it's a real struggle. And it's, again, we all have limited experience in the context of CRISPR-based medicines on both sides of the pond, but it's not any different.
I had a gentleman here with a question.
So when you look at the publicly disclosed therapeutic pipelines to date, it doesn't seem to be a lot in CRISPR-I or CRISPR-A, and I wonder if you had any thoughts on that, or if that's for the future for the field, or the potential for those mechanisms of CRISPR.
I don't think I heard you correctly. It was CRISPR what?
CRISPR.
Oh, CRISPR-I. Yeah.
I mean, I can actually say in our microbiome pipeline, we do rely on CRISPR-I and CRISPR-A. And part of that is because genome editing just looks really different in bacteria. So in mammalian cells, obviously, all we as the scientists are doing is making double-strand breaks and then relying on the cell's own machinery to heal those breaks, and that's what leads to a variety of different edits. Most bacteria do not have the non-homologous end-joining pathway. So if you make a double-strand break, you kill the bug. Now that's useful in other contexts, and we take advantage of that sometimes. But if you're trying to manipulate the genetics of a particular organism, CRISPR-I or CRISPR-A becomes really useful in that context. So it is an important part of our therapeutic pipeline.
And the base editing companies use CRISPR as a targeting mechanism without the cleavage ability. So deficient Cas9 to bring a cytosine deaminase, for example, or other base editing enzyme to the right location.
Yeah.
Thanks. So another question that sort of came up in our discussion is reliable supply of the components for CRISPR is important. What are the considerations that are top of mind when considering supply and making the decision of build versus buy? So maybe Rachel, you can start us off on that.
Sure. I mean, I think it's supply of what is the first question that we have to ask. For so many of our company's therapies, it's really complicated, right? For a gene-edited cell therapy, you probably have Cas9 protein, one or multiple guide reagents, maybe a viral vector to deliver one or multiple gene cassettes, and then ultimately the actual cell handling component to manufacture the therapy itself. And so I think every organization has to look at each of those individual pieces and understand, again, sort of the risk-benefit analysis of investing early in owning your own manufacturing capabilities. And I think Editas' recent acquisition is a really interesting example of that, compared to relying on outside vendors who have expertise in that specific area and stitching those capabilities together. I also think it's important for companies to think about what will commoditize across all of those pieces, and if anything will commoditize, over what timeline, because I think that impacts people's investment decisions in the very near term as well. And I think we benefit as a field from the fact that a lot of the components we need look a lot like a lot of the components that have been made for other therapeutic modalities previously. And so whether it's taking advantage of a lot of the chemical synthesis knowledge that comes from the RNAi or antisense oligonucleotide therapy space or other areas, we're not starting from scratch. And so I think that's a really important component of how our field has moved as quickly as it is already.
Any other comments from the panel on sourcing? We got a question, yeah? Thanks.
Quick question, picking up
Sure.
on what you just said, along with the earlier comments about lipid nanoparticles. It was fascinating to hear Coen Lab that said they're abandoning lipid nanoparticles, going to try GalNAc. I'm sorry, the microphone. So fascinating picking up on your point. Alnylam is abandoning lipid nanoparticles going to trigalnac, and I'm wondering, per your point, would it make sense to have a delivery system where you could have antigen as well as the therapeutic attached? So you get that targeting that you were referencing, Bill, and then you would get a lower dose, presumably, because you'd have more drug at the disease site. I guess that's a question.
What I think about there in terms of differences also is that's a chronic treatment versus a one-time treatment. Where I think there's, at least for me, there's less of a concern in terms of potential effects of the delivery system itself. It's not that you're not worried about it. It's just that if you're only giving it once, then it's a different problem than if you're giving it chronically.
Any other? Yeah, great.
Hi. So my question is around thinking about future modalities that are maybe not CRISPR, because CRISPR, as we know, is a bacterial system. Of course, it works great in many ways, but also has its challenges. So how are all of you thinking about future systems that we might imagine that maybe have a human basis?
So a human-based gene editing tool that we could pluck out. I mean, I think one of the remarkable things, and particularly what we've seen even in the Nobel Prizes Monday and today, is this notion that nature invents really remarkable things, and we take advantage of it. And it's really hard to speculate on, is there a human gene editing system somewhere buried inside of us? And I view our role in the companies in industrializing these inventions, be it the antibodies of the Nobel Prize or the CRISPR technology, to really figure out a way to exploit that for human benefit. So we don't spend a lot of time wondering if there's something novel or magical inside of the human cells, in part because it's hard enough just making a medicine out of complex technology, and it takes all of our focus to be able to get there. But maybe my colleagues have a different view.
One of the things that we are doing is a collaboration with Bayer and the protein engineering group that they have there. They have this fabulous group, and we're working with them to try to create I don't know if I would call them next generation Cas9s, but Cas9s nuclease systems that will have different features, and then you can imagine what the things that you might want to try to dial in and create in terms of lower immunogenicity, potentially smaller size, a number of other features. So I think there's ways of getting to what you're describing, and ultimately we'll probably will have a toolbox of tools that we'll pull out depending on the indication. Is it in vivo? Is it ex vivo? Where is it? And do you want transient delivery or not? So that's ultimately where I think we get to.
Yeah, and just to put a plug in for BD at a small company, I think, as a small company, we're focusing on exploiting CRISPR-Cas9, but we recognize that there's the potential for disruptive technologies out there, right? So I think all companies have to really not get blinded by their own platform and their own technology, and keep an eye open for other sources of innovation that are out there in the academic field and in other companies, and recognize that next generation may come through. So it may not be something that our internal folks focus on, but from a business development standpoint and a scouting standpoint, you keep an eye out for.
I think we have maybe time for one more question if there's one out there.
Well, in absence of the microphone, let me just shout. You mentioned that a lot of the innovation is crowdsourced, and it's certainly the case that the IP in the CRISPR space is, shall we say, complicated. I'm worried about, is anybody going to be able to bring a therapeutic to market with so many different competing IP claims? Can you talk a little bit about what you see as the IP milieu for CRISPR in the next 5 years?
Well, maybe I'll turn it around and talk about the product milieu for CRISPR, because I think if you have a product that is going to bring value to patients, there will be a way to get it to the market, right? So I think we are all focusing on developing products for high unmet need indications where the patients really need that, and we will find ways to bring those products forward to get them to the patients who need it.
Yeah, in the setting of unmet need, the other considerations haven't prevented products from getting to market. It's really in the setting of third, fourth, fifth to market that it's more complicated, but I think what we're trying to do is treat some pretty substantive diseases.
All right, it's gone by way too fast. I want to thank the panel here. It's been a great discussion. I'm sure you'll be around to follow up on questions and such. And then let's give everyone a round of applause.